Welcome back to the Synapse sessions. Today we are honored to host Dr. James Murray, a professor of biological sciences at Cal State East Bay. Dr. Murray is a neuroethologist whose research explores the use of sea slugs as a model to uncover the neural networks that underlie navigation, orientation, and sensory processing, linking individual identified neurons to real-world behaviors in both the laboratory and in the ocean. Beyond his scientific work, Dr. Murray is a passionate advocate for science outreach, leading initiatives such as the Bay Area Brain Bee and the Society for Neuroscience to inspire high school and college students to engage with neuroscience. Thank you, Dr. Murray, for joining us for EV Neuroscience Club's Synapse sessions.
Super dupe. Happy to be here.
The first thing I wanted to ask is, what drew you to study the brain?
That's actually kind of funny because you probably heard that like most people in college like change their major like 5 times. And what's really funny is I'm one of those few exceptions. When I was in elementary school, I enjoyed going to the library into the biology section and just pulling out books on animals. I liked animals. And we didn't have an internet, right? So the library is where you got new information. And so I just read a lot of nonfiction books about animals. And we had television shows back then called like Wild Kingdom, where they're showing animals on the Serengeti Plain, and we had the Ocean World of Jacques Cousteau, animals underwater. So I'd watch those shows, read those books, and what I realized when I was in high school is that what I really enjoyed about animals was animal behavior, right? The weird things they do. It's amazing things they do. And then, in high school, now I'm old enough to realize, oh, what causes animal behavior? It's the brain. And there are people who study the brain. Um, and I was really fortunate to be in a public high school that had great resources because we were in a suburbia during the early 80s when downtown shopping areas were being less shopped at and people were moving to big malls in suburban area. They chose our town for the biggest mall in New York State, right at the
Mm.
crossroads of a couple of interstates. And that was my town. And so we had this huge tax base. So we had all these classes. We had free AP exams. We had free tuition if you wanted to go to college in summer or at night school. And we had a free internship program from our high school. And so I said, yeah, I'm interested in the brain. What can you do? And they said, oh, we're going to send you to the University of Rochester Department of Neuroscience. You're going to be, run around with the department for a day. And they had me meet like 5 different people that day. And I got to be in the lab. I got to hold a human brain. I got to watch someone start a surgery on a bat brain. It was fascinating. And then that I really loved. They also put me in the room with a pediatric neurologist, which, I didn't know much about. But basically, I got to be fly on the wall for a week in her office. And the couple of things I learned about her is one, she was highly sought. She had appointments out years. She was such a professional and so valued that, people had her booked for years. And, who knows how much money she made. I'm sure it was plenty. Um, but as I sat in her office, I realized that she um, uh, do almost the same thing with each patient. Pediatric
Mhm.
neurologist, so a lot of teenagers, sometimes younger. The patient would come in and she'd do standard neurological exam, and she'd, ask them how they're doing. And then pretty soon after that, she'd re-up their prescription for whatever psychogenic drug they were taking. So hyperactivity disorder, things like that. And um, and it was the same thing, like 7, 8 times a day, appointment after appointment. And man, it was boring. Um, I did not find that interesting. And so working on animal research and how brains control behavior, that just, fascinated me. And the fact that the human brain was this black box that we can't really study. We can give them, drugs and see how it changes their behavior a little bit, but we really don't know what's going on. It wasn't satisfying to me. So I got less interested in medicine and more interested in kind of figuring out how animals work.
That's amazing. And that sort of leads into my next question. How would you break down your current research?
I think, the way you described it was good. Um, I started as an undergraduate researcher studying insects, and and it was interesting because we I took a neurobiology course. We had a discussion section run by a graduate student where we'd read papers, and so we'd read papers like
Hmm.
Scientific American, easy stuff. Right. And Scientific American has lots of pictures, right? And so I'm looking at this Scientific American paper with my section leader, and it was showing a cockroach and showing that cockroaches have particular neurons in their nervous system that are coded to be active when the animal is attacked from the lower right, and a different neuron that'll be activated when they're attacked from the lower left, upper right, upper left, and each of those neurons was coded for an escape behavior from,
Hmm.
how cockroaches are hard to get. And so he was showing me exactly, we figured out why cockroaches are hard to get. This neuron does this behavior, one-to-one correspondence. And that just blew my mind. And
Hmm.
at the neuroscience, at the end of the article, it says, this professor, Jeff Kamhai, is at Cornell University. I'm like, I'm at
Hmm.
Cornell University. I'm going to go find this guy like right now. And I found out he left the university like 10 years previous. So it's really sad. But I asked, is there anybody else on this campus doing stuff like this? And like, yes, there is. And I found Professor Ron Hoy, who studies basically the same thing in crickets escaping
Hmm.
from bats. And so I worked in his lab for a year and a half. That was certainly eye-opening. Um, and I really did appreciate the what experiments were necessary to show that a brain cell caused a behavior. He was very, very patient with me. He easily could have not had an undergraduate student. So, undergraduate students probably take more time than they benefit the faculty, I think, maybe, right?
Hmm.
Because he could hire grad students, postdocs, and get a lot of research done 24/7. I was an undergrad, I was doing, 2, 3, 4 hours of research a week, right? But he gave me this great project and taught me some basic things. And then the grad students and postdocs helped me as well.
Hmm.
And again, they didn't have to do that. And so I had such a great experience in this lab, learned so much, and definitely was interested, in neuroethology, right? That's what we call our field is, ethology being the study of behavior in natural settings, and neuroethology, the neural basis of that. So that's when I really focused on neuroethology. Then I went to I applied to a bunch of grad schools, got to visit a couple of them, and eventually decided on University of Washington. And they typically let you go through rotations where you'll
Hmm.
take a quarter in different labs. And so I ended up in this last lab in Dennis Willows's lab on an island at a marine station studying that sea slug. And partly the location was great, partly the principal investigator Dennis was great, and the slug model was really cool. He also had a figure that said this brain cell does this behavior.
Hmm.
It was just awesome to me. And so, my current research is, how do these brain cells cause adaptive behavior? And now that we're into, robotics and autonomous vehicles, that's kind
Hmm.
of what my research was. I was thinking would have applications in the future is, that robots still occasionally get caught in the corner and, banging into a corner and can't get out because they're not so sophisticated. Animals never have that happen to them. There's something slippery about them that, they can never like bang into a corner and never get out.
Hmm.
They're always going to get out somehow. And so that's part of my research is trying to figure out what is how is navigation accomplished. Uh, but because I'm at the California State University, I don't have to have funding all the time. Whereas, I see this is sponsored by UCSF, our Zoom session, right? If you're a scientist at UCSF, you have to have grant funding. You will not continue in your career without that. So my main job is teaching, and I'm allowed to sort of pursue whatever research I find interesting. So I branched out into magnetic orientation. I branched out into one thing about the sea slug is it's toxic
Mm-hmm.
and doesn't have any predators that can eat it. And I'm interested in physiologically how do they protect themselves from predators. So now I'm studying the predator Dungeness crab, finding out
Mm-hmm.
why they live with sea slugs but can't eat sea slugs. And so I'm branching out into that. And then I've also got some molecular colleagues that were trying to figure out how do they detoxify toxins
Mm-hmm.
because they themselves eat toxic food. And so I get to do all sorts of different research and it's really only possible at a teaching university.
Mm-hmm.
Mm.
Yeah, that's awesome. And I think one question I had when I was reading over your work was that often we focus on like a rat model or when we're looking at humans, we'll try to find a mammal that's really similar to us. What do you think that we miss when we overlook invertebrates like sea slugs?
Well, I think a good example is the 2010 Nobel Prize in Physiology and Medicine, right? That went to invertebrate research on learning and memory, right? Eric Kandel, working in the sea slug, was one of the recipients, and the reason he got the Nobel Prize is because he was focused on the sea slug with these giant brain cells, with fewer brain cells, and over a career of like 30 years he showed the neural mechanisms of short-term and long-term memory and the biochemical mechanisms and the genetic mechanisms, and then found out, lo and behold, same biochemicals, same genes used in mice and humans.
Mm-hmm.
And so the reason he was so successful over those 30 years is because he didn't start in rats. He started in the sea slug where they had a simple defined circuit, sensory neuron, interneuron, motor neuron, just three neurons. And so he spent decades studying those three neurons and how they interact, right? And
Mm-hmm.
very, very overly simple, but he learned things that were we didn't know before, were generalizable to all other species. Uh,
Mm-hmm.
and so I think that's the main thing is you have to appreciate that technically sometimes you're only capable of doing experiments in simpler critters. Uh,
Mm-hmm.
and you may know my colleague here, Dr. Siddharaman, studies Drosophila, right? And in Drosophila, they have mapped the entire brain in all of the
Mm-hmm.
synapses, right?
Yeah.
And we are not going to do that in a human brain anytime soon, or even
Mm-hmm.
in a rat brain anytime soon. Um, but that allows them to ask questions that can't be asked or answered in vertebrates.
Mm-hmm. And And on that topic of questions, the most impactful research comes from having the methods to answer those questions. And you've worked with a ton of methods, different experimental structures. I wanted to ask, what were a few that stood out to you? A couple of experimental techniques that were really interesting that you enjoyed exploring.
Yeah, so two of them that I had to learn when I was in my grad school were really, really cool. One, I learned a technique to inactivate neurons, single neurons. Or actually, I learned two techniques to do that. One, you can inject a single neuron with a fluorescent chemical and then
Mm-hmm.
illuminate it with UV light. That will create so many reactive oxidizers to kill the cell. Uh,
Mm-hmm.
and I was like, that's fascinating. Now I have a brain that used to have 6,000 cells, and now it has 5,998 cells because I killed two. And now I can figure out what are those two cells doing for the animal.
Mm-hmm.
And like I said, creating radicals in the brain is not the safest thing, and maybe has some side effects. And so I learned another technique for injecting into the brain a lidocaine derivative, right? So
Mm-hmm.
regular lidocaine you rub into your skin. Lidocaine is fat soluble, right? It'll go through
Mm-hmm.
membranes. So you have to make a new version of it that's water soluble that you can inject into a cell and then it won't escape from that cell.
Mm-hmm.
And so that time, that cell is not dead. That cell is instead incapable of generating action potentials because that's what lidocaine does. It blocks sodium channels. And so that was again a even better technique, studying how those brain cells do certain things. And
Hmm.
again, with the sea slug with only 6,000 brain cells, I was able to prove that these two neurons were essential for turning. Right.
Hmm.
So now you know they're essential for turning. Now you know this is where all the information has to go through to control turning, right? And
Yeah.
it really helps you sort of gather, a focus point.
Hmm.
The other technique that again I had to do, and again in neuroethology, a lot of our research is about proving, the role of single neurons in behavior, and so logically you want to prove that the neuron is necessary for the behavior, and
Hmm.
it would be easier if you could prove that the nerve cell was sufficient for the behavior, right? Not just needed for the behavior, but it's the only neuron, right? And that's
Hmm.
not always true, but it could be true. And so I wanted to go test uh, sufficiency as well. And in order to do that, I had to activate a single cell, which is pretty easy if you cut open the animal and you keep it in a holder and you hold electrodes in the brain. But that's not normal navigation.
Hmm.
So normal navigation crawling around in the bottom. And so I need a technique where I can record from the brain, but let the animal crawl around, which
Hmm.
I don't know if that's possible. And sure enough, I read a paper that it was possible. You can glue wires onto the brain that are thinner than human hair, and
Hmm.
the animal can crawl around like that. And so I wanted to learn that technique. I read about it, and then I heard one of that lab's graduate students just came to visit my main campus in Seattle.
Hmm.
Maybe I can convince him to come here and teach me that technique. And
Hmm.
he did. And that was also a fascinating technique. And I taught it to my grad student. We ended up publishing on that. And so those are some really cool techniques that we enjoyed.
Hmm.
But the question you didn't ask is like, what about techniques that you don't know about? And
Yeah.
that's where I'm feeling I'm feeling pretty old, right? I'm getting pretty old here and thinking about retirement, but I feel grossly ignorant about molecular biology.
Hmm.
And so I don't have a lot of training in that. And
Hmm.
so when I retire, we're allowed to phased retire in this university. And so during that phased retirement, I could teach half time and do research half time, and I might go back and learn some molecular techniques.
Awesome. Yeah. And that's a great transition to my last question about your research, which is what breakthrough do you want to see in the next 10 years as you're looking to the future of neuroethology?
For my research, finding the magnetoreceptor. That's
Hmm.
that's the thing that I'm really interested in. Um, you may not know, we have not yet found a magnetoreceptor. There have been a couple of false starts, and there's a lot of circumstantial evidence of where they are.
Hmm.
Um, and there's good evidence that there's at least two kinds.
Hmm.
And so my slug can orient to magnetic fields in the dark. So it's
Hmm.
not using the light mediated kind. And so the other kind that we know exists is uh, ferromagnetic crystals.
Hmm.
And so we know that some animals use ferromagnetic crystals to navigate because if you hit them with a really powerful magnetic field, you can dislodge all the magnetic crystals. And if they're dislodged, the animal becomes disoriented. So you can
Hmm.
literally just put them in a big magnetic field, hit them like an MRI type strength magnetic field, like whomp, big change. Rotate all of their crystals, and then they become magnetically disoriented. And I haven't done that experiment in my slug, but maybe I should. But we're not using the light mediated one. We might be using the crystal mediated one. If we're using crystals, now I have to find a cell with the crystal in it with some sensory apparatus around it.
Hmm.
That would be the thing that I'd love to find in the next 10 years. One thing we're starting to do now is we're starting to scan the entire skin surface looking for iron.
Hmm.
And that would be the start.
Awesome. And now I just want to transition to a couple of questions about your work in outreach to the community. Um, so you've been an avid advocate for neuroscience in high schools through organizations I mentioned before. and you have an upcoming initiative called the Neuro Discovery Days. So I want to ask you what your hook to students who are interested in neuroscience is. What's that thing that you think everybody should and could latch on to?
I think the thing that I try to get people started with is you know, the book by Francis Crick. He wrote a book called The Astonishing Hypothesis. And the
Hmm.
astonishing hypothesis he's referring to is that the mind is a function of the brain, just like digestion is a function of the stomach. It's not controversial, right? You can look at the stomach and figure out how digestion works, right? And we have lots of evidence that the brain is what's causing our mind. Our mind is the most complex thing in the known universe, right? I mean,
Hmm.
our mind does everything, culture, our memories, our thoughts, our loves, our hates. It's all our mind. And so
Hmm.
you can argue that all of human endeavor is neuroscience, right? Because all of human endeavor is human behavior, right?
Hmm.
And and so I like to get people started thinking about, you have a brain, and it is the most amazing thing in the known
Hmm.
universe. Wouldn't you like to know more about how it works, right? We know a lot, but what we don't know is incredible, the incredible amount
Hmm.
of stuff, right? And I was just telling my neurobiology class the other day, I'm going to spend this time explaining to you how we break down sound and turn it into detecting of words, and we're going to
Hmm.
talk about how we process light and turn it into images. And I told them that the one thing we don't have any clue is we know the brain has modular function and farms out different parts of our world to different parts of the brain. What we don't understand is how we pull it all together into one unifying experience. And this
Hmm.
is called the binding problem. Why does the world appear to be one thing when it's processed all over the brain in separate sections? And
Hmm.
and, um, and so I like to, run people with weird questions like that. I like to tell them about, the man who mistook his wife for a hat and literally took his wife's head and tried to put it on his own head, not because he was stupid or demented. He had had a focused brain damage where he could not identify items like
Hmm.
tools and things like that. Right. And so I like to get people interested in the fact that the brain is doing everything for you, and it's so sophisticated and so weird that how could you not be interested in
Hmm.
it?
And my last question to you is, what is your advice to the next generation of neuroscientists?
I guess twofold. One, everything is neuroscience, and
Yeah.
what I would say is that if you're not interested in sea slug behavior, fine. There's all sorts of stuff you can do in biochemistry, in physics, in psychology, in sociology.
Hmm.
There's so much you can study in neuroscience. My one of my colleagues told me when I told him I was a neuroscientist, like that's not a real field. And I'm like, what do you mean by that? It's not a real field. And what he meant is it's not like developmental biology where you have one approach or evolutionary biology where you're studying one process.
Hmm.
Neuroscience is this artificial conglomeration of techniques all focused on a type of tissue,
Hmm.
nervous tissue. And there's all these different approaches, molecular and behavioral and stuff like that. So I guess what I tell people is neuroscience is so broad that if you're interested in it, I'm sure you can find, something that would be specifically interesting to you. And
Hmm.
and uh, and I think it's going to be a growth field because our gross ignorance about how the brain works and the fact that how the brain works is so important for us, right? Mental illness causes so much suffering on the planet. Dementia causes so much suffering. The fact that our brain can't heal itself causes so much suffering. And so I would say, uh, I would encourage people to look into neuroscience and all the different approaches because it's such a fascinating field, and I think we will be in need of neuroscientists for at least another 500 years.
Hmm. All right. That's incredible. Thank you so much for your time today. I
Yeah,
truly enjoyed talking to you, and I really appreciate the time you've given to our club for this seminar. Thank you so much.